Ce soir je serai la plus belle pour aller danser. Danser pour mieux évincer toutes celles que tu as aimées. Aimer. Ce soir je serai la plus tendre quand tu me diras tous les mots que je veux entendre murmurés par toi.
Hi everyone, how have you been? How are you this weekend? So, exam is on next week's Friday. So, before we get into the next chapter, which I'm going to cover approximately about half of the actual entire chapter for today, and that will be the the end of the material that I'm going to cover for the upcoming midterm exam. So the midterm exam will cover from the start of the whole lecture in the semester up until what I get to finish today is going to be the extent of the material for the exam. Okay, so as all of you hopefully are aware of by now, the midterm exam will be on next Friday, starting from 5 to 8 p.m. And the the way the exam will be taken, we are using the Zoom online conferencing program. So everybody should log in to the Zoom conference, the login information that I sent, and then you can just follow the link. So for that matter, we are going to have another recitation session during which, if you had any questions, then we can go over the specific question regarding some material concerning the exam. And so that will be on Wednesday at 5 p.m. We don't know how long it will last. I kind of anticipated about an hour and a half. Like I put that information on the announcement. If for some reason, if you can make that recitation session, then would like to still like have participated in, then the entire session will be recorded so that you can later, maybe the day after, you can try to review what has been covered, sort of things. And also, even during the recitation session, even though you can have questions in Korean, as you feel, probably perhaps some of you feel more comfortable the way that is, but I am compelled to reply as much as I can in English, because this, for one, this is absolutely an English speaking lecture class. That is the original design of this lecture course. And another thing is, there are several international students who are not in this classroom, who are not native Korean language speakers. So even though I would like to, of course, I feel more comfortable speaking and communicating in Korean too, but that is not something that I should refrain from. So please understand that. And also, during the exam, same thing. I emphasized several times during the recitation before, and also the start of the lecture in the semester during the class orientation. But during this exam session, especially during the Zoom session, you will be asked to keep turning on your camera on, so that at least I can check on the eyeballs and your face. Although the ideal scenario is actually you can direct one of the camera to your side view, so that I can both monitor your screen as well as your activity. But I wouldn't really do that much of the specifics because this is not something like a Samsung Corporation, what if the admission exam type. So I don't really have to do that, but still I can check on the most of the your activities that I can pick on. If there is any unfortunate thing or activity was going to happen, then I can at least try to prevent in advance. So other than that, actually, you will be pretty much flexible while you are taking the exam. So make yourself as comfortable as you can, including that you can the place of the exam taking could be anywhere, like as you please. So you can take the exam wherever you want. You feel comfortable. So even you can actually take your exam in the cafeteria. I don't know. In these days, the cafeteria will allow you to do that or not, due to all those restrictions. But anyway, if you can, but still, the one condition is you should be able to turn your camera on and then face towards you. That is only one condition. And also during the exam, like you can do whatever you want to make your body actually physical body comfortable enough so that you can drink and you can eat, and so it's going to be a long exam. It's not that that exam will take that much amount of the time, as I said earlier, but still, like it's just the idea of to give you the ample time so that the time limit should not be any excuse for your poor performance. Is the idea of giving you 3 hour long exam. But that 3 hour long exam is like almost a torture. So that's why you can be as flexible as you can and comfortable. Also, as many as you want. You can leave. You can take breaks during the exam. Yeah, that's another kind of a unorthodox way of taking the exam. You don't really actually imagine that in any other exam environment that you could do that. But I've been allowing students to do so, and no problem has ever occurred as far as I know. So you can leave the site to take a break whenever you want and come back. How long it will take, it doesn't really matter because the exam is whatever. It's just the entire during the 3 hour exam. You can take any breaks that will those break times will be subtracted from your thing. So it's your like if you take too long break, then you will be the one actually who have to deal with those loss of the time. So I don't really have to worry too much about it. But the only thing is that you have to leave your cell phone on the site, so that you don't really want to take your cell phone with you when you go out of this exam site. That's the only thing. So you just make sure that I notice that you are leaving your phone on wherever, like on the table, or like at least you're not taking, to make sure that you're not taking your phone with you is the thing that I want to check. So the exam, actual physical exam, in the form of a PDF file, will be placed on the on Snowball class announcement section. As I said earlier, 15 minutes prior to the actual start of the exam. So that will be 4:45 p.m. Approximately. The reason that I am actually putting up the exam that much early is so that if you can afford to have the printer nearby, then you can have like a time to prepare, get the exam printed out, so that the conventional way of taking the exam. Some feel that it's much more comfortable to have those paper exam and then write down on the paper. So yeah, so be it. And if you are like, unfortunately, if you don't have any access to the printer, don't you worry, but simply you can use the actual screen of your whatever, your phone or your computer, like, and then just write down only the answer you can. So when you're finished, like I said, you have maximum 3 hours to finish. Then you have several options in turning the exam in to me. So one of the easiest way, probably, perhaps, I guess, is to take the photograph. If you are actually using the paper exam after the exam get printed out, then you can take the photograph of that exam with the answer on it, and then send the photo file directly to through email to me. Or you can, if you are a little bit of a skillful, so that you can, if you can convert them on your own into the PDF file, that will be also nice way of doing so. So just either way, so then you can just send it to me. And also that when you don't have any printout exam, just write your answers on a sheet of paper, or even only write your answers on the word processor program, anything like a Word or Apple Pages, or even some people prefer using the Hangul. Personally, I hate that program, but anyway, it doesn't really matter. So you get the idea, hopefully. So there are several ways of sending the finished answers. You can then have them with the actual original the questions in the form of an integrated PDF file, or integrated the photo file, or the answer only computer files, or answer only photo files. It doesn't really matter. Okay, so that's what it is. So before, like, so from today to up until the actual next Friday, in the meantime, if you came up with any kind of concerns or questions about this procedure, exam process, then just let me know. Okay. And what else? Oh, the actual turning in the exam. Also, that's another. Some people have the questions about it. You can have entirely full 3 hours of working on the exam. So after you finish, turning in process is something extra gravy. So it doesn't get to be included in that original 3 hour exam time. So you just use up all 3 hours, and then you still have a 30-minute maximum, 30-minute extra, during which your exam file should arrive in my email box. Okay, that's 30 minutes. I think this is really generous amount of time, so that during which you can work on. Sometimes the email, when you try to send the email to me, it doesn't initially get through very well. So then you try to fix the problem. If you cannot fix it on your own, you can just have a communication, constant communication with me as well, so that basically that after the exam, during that 30-minute extra time period, every exam should arrive in to me. That's the idea. Okay. So other than that, we can get into the new material, which is about the cell structure. So here in this chapter, the purpose is we are going to look at internal structure of the cell, so that understand its different components within the cell, and their functions. We will start surveying the prokaryote first, but actually the main goal of this chapter 4 is to look at the eukaryotic cell, because eukaryotes are the one actually that eukaryotes distinguish themselves from prokaryote by having so many different types of internal structures. We label them, recognize as organelles. Unfortunately, prokaryotes don't have them. Okay, maybe I don't know. Really, actually, I should label that as unfortunate. Maybe that's their blessing. Who knows? But anyway, so before we get into the actually the viewing and then looking into those internal structure of the cell, then we needed some kind of a tool, scientific, technical tools. So how do we see the visualizing cells has been the problem for a long time until very recently. So this is one very nice real photograph of so many different microorganisms. All of them appear to be some type of different forms of bacteria, and these are not artistic representations, but some particular type of a photograph taken using some specific type of microscope. So let's get into first that several tools in the form of a microscope which enables us to survey the small world of these cells. So microscope, actually literally like a microscope is an instrument, but the invention of this microscope ever since then has been the instrumental, very helpful in understanding, deciphering the whole different world of cells. This is one of the small, handy, so to speak, that is not really big, sophisticated relatively speaking, microscope, but even these are really very fancy modern microscope. So in using the microscope, we can actually visualize a very small, fine scale of cells. So if we, taking this example, let's trying to zoom in on our certain area on palm. So animal cell. So let's start with if we just zoom in, and then if we reach something like a 10x magnification, you will be able to see this. And now we can somehow magnify this area 100 times enlarged. Now, and then if you go into further, like for example, like 1,000x, now just totally whole new different world actually unfolded to you. Look at this. You have never noticed this type of things ever actually in your palm. And look at even the 10,000x magnification. Now it's time that you actually are able to distinguish one small, fine, detailed component of a single cell. So inside of this single cell, how do we actually distinguish each different, all these different small components, and can you know what they are and then what are their functions? And so those information accumulated, so then available for us these days, have been the tens of thousands of like all those marvelous hard-working result of such hard works from all those previous scientists using thanks to the invention of this wonderful tool device we call the microscope. It began in something like in 1600 or something. Robert Hooke, although he was not the very first person who actually invented this type of so-called microscope, but still, generally, this gentleman is regarded as a pioneer of the light microscopy. Um, so this is perhaps this must be a replica. I don't think this is actually original microscope he invented, the original form. But the idea is using this. This is the image of the first type of specimen he was able to look at. It's actually plant cells at cork. Uh, The cork cells are the outside covering of dead plant cell, so it shouldn't be actually regarded as a real cells. Nonetheless, his achievement by recognizing this for the first time was something really monumental. So anyway, the first invention of his, the microscope we these days we call, was some type of the compound microscope having two double lenses. There have been some of the other types of microscope those days having only one single lens. Still, with that single lens, that microscope also could achieve a greater magnifying power. But anyway, he was able to up to something like 30 times magnifying powers having microscope. And then with that, he was able to observe these plant cork cells. And the thing is, through his eyes, it looks like something like porous structures. So his description, according to his description, is it looks like a honeycomb. And then he, for the first time, that type of structure as a cell. Okay. So that's why, even though that actual cork cells are not a living cell but a dead debris of the plant cell, but still, it is something very significant. The first step for us, the human, now actually correctly approaching the study of the cell ever since. And this is his original publication regarding his the observation of these cells. Okay, so these days, so-called this limit, there are two different parameters when you try to handle this microscope, and how much can we see, how much magnified the image can we get through this microscope, so to speak, is the magnification power is up to about 1000x. It's a normal light microscope, regular light microscope. There are different types of microscopes, so it is specifically only about the standard light microscope, which that utilize the visible light. All those light microscope use the visible light. There are in the light, some portion of this light are not visible to our eyes, like ultraviolets, wavelengths, and also far red and infrared type of wavelengths of light. We are actually usually it's very difficult for us to recognize. So only those like, so to speak, rainbow color wavelengths of light, we call that as a visible light. And in light microscope, we need this source of light so that we can take a look at any sample. The sample we call the specimen. So how much can we enlarge any using this type of strategy, enlarge any specimen? It's about 1,000 times. It's a lot, 1,000 times. You have already seen this when we actually magnify our one spot on our palm 1,000x. At least you can see something is going on inside, like a several clusters of cells. You are able to distinguish. Okay, then if you go up 10 times or further, like 10,000x, then now you actually get to distinguish one single cell, and even you can see some of the structure inside of the cell. So that's the magnifying power. However, another parameter is the something called resolution. This resolution is, as being said, the resolution limit of modern light microscope is something about 0.2 micrometer. Micrometer is a 1 millionth of a meter. So 1 micrometer is extremely small, actually small scale, small whatever the length. Okay, even then it's 0.2, not even 1 micrometer. So resolution is 0.2 micrometer is something that really pretty good resolution actually. However, that is not really good enough to distinguish every bit of different components in the cell. So this is one the rational logic of how the light microscope works. So here is our observation spot, our eyes. And then if you place only one lens, a single lens, this is a simple microscope, then you get to this when you place in an object underneath of this one single lens, then lights passing through this lens will bend as images. So that's how you can get this. Originally, the size of this object is something like this large. However, using this lens, you can get this much enlarged image. But if you decide to use two such lens, then this the bending of light, actually technically we call such thing as a refraction, right? This refraction of light, because you have these two lenses, and then the image will be like inverted. You will have some kind of inverted image to your eyes, but other than that, you can enjoy a greater amount of this magnification using these two lenses. And this type of having two lenses system in a microscope is called a compound microscope. Yes. And this is about the resolution. So resolution is if you magnify an object to a certain limit, then you get to reach that, so that these two gap, because it will get further and further magnified, so then these two gap cannot be distinguished any longer. So that is where you have reached to a certain resolution limit of that particular device. That's what we call the resolution. And that resolution can be described as this type of formula. Of course, you do not have to worry about memorizing and anything about. The only thing that I throw this type of mathematical formula is to give you what kind of parameter determine this so-called resolution. Just like your TV set. Before, when in 1980s, all those TVs in our home was something that having very poor resolution. That means the size of a pixel on the screen is very large, so you could not really distinguish any like the fine images. So the image could be like to today's standard. All those images were very blurry. Resolution is very not good, not very good. Same thing can be applied to the concept of microscope. So the higher the resolution, which means this d value becomes smaller, which means this. Further, you can actually much smaller gap can still be distinguished if you have higher resolution. So how can you reduce this d value so that overall resolution is increased? One way is you mess around with this lambda value, okay? But this lambda value is a light wavelength, so there is only so much you can do about this. And alternatively, you can try to increase this component, but there is also so much you can do about this. Numerical aperture is something is being decided by what kind of a medium, like where you are visualizing this material in through the air or through the oil. That's why actually, if you are some of the for those of the majors like bioscience major or those science major must have taken the laboratory, the college or high school level laboratory classroom, and when you using the basic light microscope, when you are doing this laboratory exercise, if you want to go up to higher magnification of that light microscope, something like 100x, okay, on the visible part, then you should apply this oil. That's the reason why through the oil that you can have a little bit of the increase of this value so that you can increase the resolution. That is the reason why you were told, probably you must have been told, that when you go up to 100x, you have to apply small oil, and then that procedure, that practice is called oil immersion. And this is the theoretical basis for that putting up that oil, whatever. So that is one big limitation of the light microscope, this resolution. And another limitation here is you have a problem because most of these light the cellular components are penetrating passing the light through, so it's very difficult for to our eyes to distinguish clearly any such structures. So we have to rely on this staining so that increase the contrast and then put some different colors. Right? So that is the purpose of staining. But by doing so, you have to, if you want to stain your cell specimen, then you have to kill, because the staining procedure involves, this procedure is something called fixation, so that don't move. All those structures are going to be fixed. And then that procedure will actually inevitably kill the cell. In other words, you cannot visualize any live components of the cells. You cannot observe any living cell without. So like, if you do not apply any stain, yes, you can observe any living cell. So if the purpose of visualizing a sample under the microscope is simply enjoying what's moving in, oh, there are a lot of creatures moving around, just like the earlier days of scientists have done, yeah, then that's fine. However, mostly in these days, your purpose is not something just like those for fun only. You want to see actually what they are, and with a higher magnification. In that case, it is impossible. So that's one of the serious the limitation of using the light microscope. Then there comes another type of the variation of a light microscope. So this fluorescence microscope is a variant of light microscope, but in the fluorescence microscope, instead of using just visible light as the light source, they use some specific wavelengths as well laser. The reason is just simply to have some specific wavelengths of this fluorescence. What is fluorescence? Fluorescence is simply put, not everything can have this type of magic called fluorescence. Although we are like in our everyday lives, we have so many different types of fluorescence lights around us. But in principle, fluorescence means that any particles or material that absorbs light in a specific wavelength and then emit. So this is something absorb and emit the light. But when it absorbs the light, it has to absorb then specific wavelengths. Okay. Then emit, when it emits that light, once absorbed, emit to the other, and it has to in the form of some specific wavelengths which is different. Usually because these light wavelengths also involve some different degree of energy, so to speak. If you consider that the rainbow color, as we go towards the blue area, it gets the higher amount of energy, so that violet and blue lights, violet and blue portion of the lights contains the highest amount of the energy, whereas the reds towards the red and orange portion of the light contains the lower amount of energy. So in this fluorescence, probably because it also has to, this fluorescence process also has to obey the first law of thermodynamics. You cannot create any new energy out of nowhere. If you receive this much amount, then you emit this energy, then the lower than the amount of what you have originally received. So according to that rule, usually when you absorb this fluorescence in a higher, like the blue portion of the wavelengths having higher energy, then you get to emit that energy in the lower form of energy wavelength, something like a green or the red. So that's the basic rule of this fluorescence. And some of the such chemicals, depending on their structure, usually some specific ring forms of organic chemicals can do so. So they are fluorescent. So those fluorescent chemicals can do this, like emission of a fluorescence light through this type of procedure, and taking advantage of these things, and using some specific laser, then we can detect some of the fluorescence material. How do we generate some fluorescence material? Well, in nature, some materials are naturally fluorescent. We call that autofluorescent. Autofluorescent. Automatically, they are fluorescent once they have received some specific wavelength of the light, and then will emit some other very specific form of wavelengths of light, and then we call it as an autofluorescent. Plant leaves are autofluorescent. If you put the plant leaf under the fluorescence microscope or UV light, whatever, then you will be able to see some of the specific, usually it's a red fluorescence due to that pigment present in the chloroplast of the plant. That pigment is autofluorescent. So many natural molecules are autofluorescent, but also we can create fluorescent material by tagging some fluorescence chemical to some very specific molecule of our targets. So this is usually mostly the majority of this visualization through fluorescence microscope is done. This is the way. So by tagging some individual molecules with this fluorescence marker. These fluorescent markers are the fluorescent chemical, and we call that as a fluorophore. Fluorophore is the name of the chemical that can become fluorescent. Then we can attach this specific color of fluorophore to any specific target molecule, and then we can, why do you want to do that? We can follow them. Once we have done that, then we can follow those molecules in the living cell. It's just the tremendous power of using fluorescence microscope. So here three different ways of generating such a fluorescent tagging. So for example, like in the case of usually actin is one of the most common structural protein inside of both plant and animals and fungal cells, all those eukaryotic cells internal. So here in this fluorescent microscope photograph, it's a fluorograph usually we call. The green thing, all these green things are something that actin proteins that are labeled. How do we visualize this actin protein? Using the specific protein that specifically bind to the actin, so that molecule that targets actin. Then we attach this fluorophore to this molecule so that we can specifically detect, follow wherever the actins are present. And then it turns out that, each single animal cell we have plenty of. What are those blue spots in the middle? They are nucleus. There is another specific dye, fluorescent dye, specifically stain the nucleus. So we can easily distinguish where are those nucleus in a cell. So in this way, we can distinguish at least. And here, using fluorescence microscope is the goal is not simply viewing and fine structure of different cellular components of the cell, but specifically following one target of your interest. And so many occasions, actually, that's something that you need to do. And here, another thing is, this is an kind of a diagram representing any general antibody. Antibodies usually shaped like that. These antibodies are actually hung upside down. This is the base of an antibody, and these two areas are the area where this particular antibody molecule can grab intact to the antigen binding site, these two areas. But anyway, this particular antibodies can recognize a very specific molecule, right? So many different antibody, each of which distinguish and detect specifically to a certain target molecule. So using the antibody, if we put this fluorophore to a specific antibody, that means we can follow a specific target molecule using this antibody. Wherever that molecule goes, this fluorophore will follow because that antibody must have captured and then bound. So that's another way of a very popular way of generating such fluorescent tag. And the end result is you can generate this green fluorescence of your lectures. So whatever it is, you can actually follow that molecule in the cell. And also, in generally, if you also have this fluorophore attached to this particular chemical, then you can specifically label all those different types of carbohydrates and lipids. So in this way, you can also generate the specific carbohydrates or specific lipid specific fluorograph in the microscope. So you have so many. There is almost a limitless application examples that using this fluorescence microscope. And the best is yet to come. And this is another natural fluorescent protein. Its name is GFP, standing for green fluorescent protein. So this GFP protein is shaped like some kind of a barrel shaped protein. It's an autofluorescence, and originally it was taken from this jellyfish. Jellyfish glow fluorescence due to the presence of this green fluorescence protein in their body. So we humans store the gene making this green fluorescence protein from jellyfish without having any thank you note, obviously, and then decided to use in our many different occasions the purpose to visualize our own this sample. How? Oh, you can have this kind of fusion in the gene level. So let's say this area is in your gene, this area of the gene segment of the gene, let's label as your favorite gene, YFG. It is not anything green fluorescence, but your favorite gene. And you make a fusion of the GFP protein like this. So this is kind of you have just generated as a kind of a chimera, like hybrid chimera gene, and then make this chimera gene in the cell. And then now your favorite gene is going to be expressed and will make the protein in the form of this fusion. So wherever this your favorite gene protein goes, the green fluorescence protein will go together, because now this is like together. Both of them are like one part body of the protein in this way, and we have we can make sure that there is techniques that even with this type of fusion, you don't really have any negative effect on the function of this original YFG, so you don't really worry about things. So you are actually following the original YFG proteins, where they go, and then what would be their respective function there. Type of the studies can be made using this type of the GFP fusion protein. So this is much more convenient and powerful than simply using some other types of fluorescence tagging things. So it's become so popular and also have contributed to tremendously to the advancement of this whole the science in general. I myself have used several occasions this GF fusion, and then look up my own the research project, my own protein of interest under the fluorescence microscope. So actually, I can bring up my own data of this GF fusion fluorescence microscope data, even if I wanted to. So that much is. And see, this one is a structural thing. Is then internal inside of this yellow thing is actually the floor of for responsible for this lighting up the green thing. It's beautiful. Maybe only to me. So. And now using such GFP, look at this. What are this? What a disgusting things. Whatever. What have you done to these poor mice? Some mouse having so-called transgenic mouse, like something like GMO which is equal to GMO. Yes, this mouse is obviously expressing the foreign gene of the GFP fusion version of any protein, and that protein must have been something that is being made. I mean, the gene is expressing everywhere, on at least on the skin, and so now it's being expressed. You can actually detect on your eyes. And these are the ones actually not expressing such gene, so they don't glow out in the dark like under the UV light. And this particular mouse, in the case that the GFP protein fusion must have been done in different type of protein that is being only expressed in this particular area of their body and tissues. That type of distinction you can follow using this. But in reality, actually, there are so much more, and then the greater details can you apply these techniques of GFP fluorescence. And people will never stop there. This is an example of such that, so that originally this GFP was this color, but people have generated color variant of different version of the GFP, so that now in these days you can enjoy whole different repertoire of more than rainbow coloration of this same GFP protein. So that, in other words, in a single specimen, I mean in a single micrograph, you can detect at a time, you can detect several multiple different types of protein at a time. Is the advantage by using different such coloration. So no wonder why eventually in the year 2008, these three gentlemen and scientists, maybe I should I say gentleman and scholar, two of them from the Japanese ethnic origin, they are really good at in these type of technical things. The whole, I don't know, that's their DNA. And so these three scientists have been recognized for their contribution in. So imagine, you may actually think this is only, it's just a simple technique, like a GFP, who cares about? But then the fact that even those who invented this technique of using GFP protein in the fluorescence microscope have been, like something like 10 years later, have been finally recognized as their contribution to the science, and then even became a Nobel laureate. Is something. It means something. Although one of these scientists no longer with us, who passed away several years ago. So, using such fluorescence technique, you can generate so many different and beautiful images. Like this is not a broccoli, but one in the part of our kidney, some specific type of a tissue cells, so only specifically labeled with this green fluorescence. Then you can distinguish and visualize such of them. And in the cellular level, you can also distinguish different type of cells like that. And on the other hand, using different type of fluorescence labeling, you can see and distinguishes many different types of cells in a single worm. This worm is, by the way, is a really very important worm in science. It's called the C. elegans. Its scientific name is Cinorhabditis elegans, and it's one of the such model organism because it has only very limited number of cells, and each of which we can follow their lineage. So it's really ideal for studying the development of animals. And this is another like a dividing cell. Now this cell is trying to produce gamete. How do I know? Because this cell is dividing into two and eventually trying to produce 4 different cells. And so this, at least this must be a gamete production. I don't know about this. This maybe it could be such a normal cell production. Like I mentioned earlier briefly, and also we are going to get it in a little more detail in the later chapter. There are 2 different types of cell. This is a gamete, a sex cell, and the normal body cells. The mode of cell divisions are a little different. But anyway, during the cell division, here the idea is you can specifically look at this. Each individual chromosomes you can distinguish in this blue color. And what are these gold things? All these golden things are the proteins used in pulling up the chromosomes during the cell division. Its name we are going to reveal later. And these are some area of the plant cell using this different fluorescence color and beautiful, and also you can distinguish different types, and also especially the periphery of different plant cell, which is something different from animal cells. And the backside of the plant leaf, plants carry usually this type of interesting looking hair cell, and those hair cells specifically can be labeled using this fluorescence microscope. So many different examples like that. So the beauty of this fluorescence microscope, not only that, it also increased the resolution limit of this visualization by using a little more advanced and sophisticated technology. So now, originally the light microscope resolution limit was only 0.2 micrometer. Now it became up to something like 20 nanometer, which is really small. That if there was something like 20 nanometer, that is something almost about the size of the individual ribosome. It's just amazing that. So here is an example of how far we have come. This was like when you try to look at this area, or the same area of the cell. Here first is the original conventional resolution limit of the microscope. Then it will be like blurry and fuzzy. So it is not really much of an help using this type of imagery. However, in these days, you can have this so much increased resolution. So now you can see on this area, you can see so many different areas of the cells can be distinguished, so that you can individually work on it. And besides, also you can apply some of the different labeling, color labeling of the same area like that. So that is how far we have actually come. This is it's also like super resolution fluorescence microscope example. So some of the using conventional fluorescence microscope, you get to only have this much of a poor resolution, but in these days you can have this much higher, clean, crisp resolution. By the way, this clathrin is some of the structural protein. At this point, we don't really have to worry about its name and functions, but there are so many different types of such structural proteins, and that working some specific function inside of the eukaryotic cells. And that is the reason why we want to have this particular chapter inside of the cells, especially in eukaryotes, what's going on, sort of things. So, and another thing is, oops, it's been already an hour. Oh my gosh, how am I going to be able to even finish this whole that I have originally intended? Let's hurry up then. Jesus. So then, among those different fluorescence microscope, something called the confocal fluorescence microscope also provides another the dimension of this microscopy. So that fluorescence microscope, but also can generate a little bit of a 3D like feeling images. How? Let's take a look at this movie so that how we generate. It's just like when you take your CT image scanning, what you do is like you have a slice of all different images, and then computer combine those each piece of a sliced image to have a whole completed 3D body image. And under the same principle, you can try this slicing of the image in the case of a fluorescence microscope. And let's take a look at this is such an example. So each different slices of image now then can be piled up to have some depths. Now you have some depths of the images. So not only unlike in the case of a conventional microscope or scope where you can have only very flat two-dimensional image view, now on this type of a confocal microscope you have a certain degree of the 3D information of an internal cellular structure. And so this is one such example of confocal micrograph, and hopefully you can see some of the some differences like in depths. So this is how you can. So it has such a much clearer and a fine resolution of the image, but also you can have a little idea about in depth of this structure too. So it's been very popular. And so that's about the fluorescence image. But something that you should not forget is the beauty of this fluorescence image. It's not something, resolution is one thing and all those the beautiful color is another, but the most significant the feature about the value of this fluorescence microscope compared with any other types of microscope is this live imaging. So you can deal with the living cell, and inside of the living cell you can follow some of your specific target molecule is such a powerful the contribution of this fluorescence microscope, and that is the reason why the fluorescence microscope is so popular in these days. So here and there, even you are not something that you are related in this science in these days, in even the newspaper or somewhere else, any other media, at least once or twice you must have seen such different fluorescence colored images, and then you might have wondered, what are those? How do they actually generate such different colors? Yeah, using such fluorescence technique is the answer. Now, this electron microscope is another chapter. Actually, originally this electron microscope has been invented way earlier, much earlier than the fluorescence microscope. So almost actually this electron microscope towards the mid 1990s or something. Personally, even I thought actually this electron microscope technology is going to be some kind of a dead dinosaur. It's an outdated techniques, although long time ago it was very hard. So when I was an undergraduate, actually I had to take the class of this electron microscope skill. So I know actually how to prepare such tedious sample specimen for the electron microscope, and I also know how to operate electron microscope. But then, since then, I didn't have any occasion to actually use any electron microscope. It's not because there wasn't any equipment available. No, it's not. Although electron microscope is extremely expensive equipment, so not many people can have an easy access to. But that was not the reason. But I didn't have any reason to use. There is no usage. So anyway, let's go over the basic principle and how they are different from the conventional light microscope. So one big difference is here. Instead of a visible light, they use this. The electron beams. Electron beams replace the light source. So it's not something, since the electron beam is the light source, our bare eyes have no use for visualization. So some kind of computer screen will take the role of our the eyes. So the electron beams will generate certain specific images, and then there is a detector. So this is one such typical, not too fancy, but typical electron microscope photograph. So basic principles are very similar to that of the light microscope, but the only difference is here in the case of electron microscope, electron beams is shooting some kind of the so-called visualization source, and instead of this lens, here in the light microscope there are lenses, but instead of that there are some of the magnet, because it's an electrons. So it's not light, so it's an electron, so it's a magnet will take the role of such lenses in the case of electron microscope. So you can have a better comparison of the light microscope versus the electron microscope. So here one big difference is magnetic lenses. It's not an optical lens, but magnetic lenses are usually multiples of these magnetic lenses are there inserted to process the magnifying and image processing until like the detector. Here the detector is our eyes, but there is a different type of detector. And the thing is, if you use this electron microscope, the magnification is great, million times. Remember that even using the highly sophisticated fluorescence microscope, the magnification is something only 10,000 or anything like that, but million times of magnification. Can you believe it? And the resolution is also extremely. That resolution, even if the highest sophisticated, most advanced fluorescence microscope, what did I say about the resolution limit? 20 nanometer. But not even tens or twenties, but it's a single digit nanometer scales of resolution can be achieved by electron microscope. Why? Because that is actually one of the reasons why actually through this the formula. According to this formula, this wavelength portion, if you replace this wavelength portion with the electrons, depending on different voltage of electrons, now this lambda value can be reduced this much. That's how you are able to reduce the resolution. I mean, I shouldn't say reduce the resolution, increase the resolution. If you have a smaller d value, then you have a higher resolution. That's why. Okay. So, so using this electron microscope, like small particles like viruses, the nasty coronavirus and the bacteria infecting viruses, all kind of different virus particles. You cannot visualize any virus particles because they are much smaller than a cell, individual cell. So it's difficult. You cannot really visualize using the conventional light microscope. Even the fluorescence microscope you cannot. But this electron microscope, can you visualize this and some other fine details of the cellular structures, including ribosomes and cell membrane fine structures and something called the cytoskeleton that we are going to take a look at later on this chapter. And even single molecules of DNA can be seen under the electron microscope. So this is such virus. The bacteria infecting virus is named bacteriophage. Virus is actually captured under the electron microscope. And this is a mouse part of an insect. Look gross or look fantastic, depending on which side you are in. So this type of detailed structure can be visualized using the electron microscope. However, the short side of this electron microscope is this. One biggest problem is it's very tedious. What is tedious? To prepare samples suitable for the electron microscope examination is extremely difficult, and it takes really skilled techniques. And also artifacts is another problem, because the way you prepare this sample, and you inevitably invite all those non-real actually artifacts. So oftentimes you get to wonder the type of image that I am seeing under this electron microscope, is it really real? Kind of you get to wonder. Is those two biggest problem. Although the third disadvantage was that electron microscope cannot generate any color image. It always has to be something like black and white image because of the specific characteristic of the sample preparation. You have to actually shoot all those metals and cover the specimen with the different metals. So there's no way you can have these colorations. But that is no longer the case in these days of a computerized imaging process. So anyway, two different types of electron microscopes are there. One is called transmission electron microscope, and the other thing is scanning electron microscope. The same samples are examined using one with the transmission electron microscope and with another with the scanning electron microscope. So hopefully you see immediately how they are different, how the transmission electron microscope is different from the scanning electron microscope. Can you see? So this transmission electron microscope, abbreviated as TEM, here the samples gets to be penetrated by these electron beams. In other words, if you are going to visualize examine any specimen under the transmission electron microscope, your sample must be very thin, extremely thin. That's another technical difficulty of you have to generate the same thin slice of this sample, like how thin? Something like zero point 0.1 micron in depth, or the thickest possible ever allowed is 1 micron. 1 micron sample is very thick, so that electron beams can penetrate easily. But the beauty is once you succeeded in generating such thin sections, then now you can reveal so much fine details of the internal cellular structures like that. So that this is one such example of like a cross section of a cell, eukaryotic cell. Here, the area of a nucleus. This is a nucleus, N nucleus, and the dark area, there are some of the holes, nuclear pore. We are going to actually look at individual these pores. And here, some specific organelle, endoplasmic reticulum. At this point, who cares? You don't really have to worry about. But all these different such structures are like visualized like this. So you will be actually surprised in how detailed the structures can be seen on the such electron microscope, in particular transmission electron microscope. So you have this type of magnification and fine details. On the other hand, the scanning electron microscope, electron beams does not penetrate because the sample is just thick, but scan the surface. And the secondary, the electron particles are detected, so that later all together, so whole surface image in 3D can be generated. So the 3D image is the key here. Scanning electron microscope can generate this three-dimensional structure. So here, a cell, animal cell, like is hung on the certain hole of this is a filter paper hole, and this is a surreal structural thing of cells and other objects can be seen on the scanning electron microscope. Another example, look at this. What do you think these are? They are all these different pollens. See, you can see different structure and shapes of all these different types of pollens there. And as you can see here, and these are red blood cell, and these are white blood cell. And like I said, no longer in these days actually you worry about the black and white imaging, because the computer convert any of those black and white original electron micrograph image into these beautiful real-like colors, so you do not worry about it. And also there's the reason why I am bringing up this electron microscope is not the thing of this type of past history, although electron microscope is great at providing you such a fine detailed higher mag and great resolution like this, but that is not the reason. Okay, this is the real reason why I want to actually introduce you this electron microscope, the cryo-EM, the new things. So it's a new development out of the conventional electron microscope. Okay, and what's special about this cryo-EM is that the structures is the key thing. Why in biology we are so much excited about knowing the structure? Once again, because once you know the structure, you can guess the function. Structure and function relationship is extremely highly correlated and important. So we can have an information about the structure and as a result the function of these small molecules using this cryo-EM. By the way, this cryo-EM stands for the cryo, which is something the frozen cryo electron microscope. So you are visualizing a sample at extremely low temperature is the the difference in cryo-EM. So at a lower temperature, you actually freeze instantly that specimen so that you make those, each different molecule make not able to move, so that you have easy opportunity to capture their instant structure. So that's the kind of a very, very rough principle, although we do not really have to go into any kind of technical details. Then the thing is, so using this technique, you can have this structural information. What's more important is, yeah, you can also have the structural information in the other way, in using some other more established conventional method. But here, one very big advantage, big and important advantage is this native state. You can get those information in their native state. Is the beauty of cryo-EM. So here comes the comparison. This so-called X-ray crystallography is such conventional method of deciphering any molecule's structure. So you just shoot the beam of X-ray to the object, and then electron beams and X-ray beams will get the scatter pattern on those detector. Like detector is something like your X-ray film when you have this X-ray examination on your health exam. Then you can have this information and then reconstruct the structural form. But there are a lot of those limitations in applying. So in techniques, so you cannot apply this technique to every different type of molecule. So that has been such a huge serious bottleneck of these techniques. But then here in cryo-electron microscope, any molecule can be applied, and then this image of structural image provide actual more realistic natural image, natural structure, as opposed to here. This is something could be very artificial and structured. Structure. Do you mean by if you know what I mean by that? So these are some of the summary of how we generate this sample under this technique, which we don't really have to go into any details, but simply the key thing is something called a vitrification. It's a freezing quickly so that even those samples become some glass-like a crystal-like glasses, or either way around. So much lower, quickly, sudden drop in the temperature generates such type of structures. And then you, using such sample, you have an opportunity to generate so many different angles of the structure. And then later, with the help of a computer, now you have a whole reconstitution of the structural info is how you do that. So in these days, actually, so many different institutions already equipped this the cryo-EM, but the Britain's MRC laboratory is the mecca of such cryo-EM techniques, and no wonder why. So several years ago, very recently, so these three scientists have been eventually been rewarded by this Nobel Prize award, and this Richard Henderson is from MRC laboratory, as you can see. And so the progress of these techniques has been so impressive in that, like even the year 2013, the resolution was this type of fuzzy, but now the present day's resolution was even at the atomic resolution scale. So last year, actually, there was that breakthrough was made, so that this using cryo-EM atomic resolution has been achieved. So here you will, as you for those of you like just you have such a long life to go, unlike me, and you will get to hear a lot of those breakthrough and achievement made by this cryo EM. So that's why I like really intended to introduce this cryo EM, even though it may be a little bit of two techniques, but maybe a couple of years later, this cryo EM thing is not really two techniques anymore. So everybody actually gets to hear, just like we get to hear the PCR or RT-PCR. So every day, just thanks to that coronavirus things. Yes. So is this? That's what I believe. So this is the fine detailed structure of the corona, that nasty bloody coronavirus. But this was also made by basically the cryo EM. So we have an information specifically about what type of different proteins are on the surface of this coronavirus, so that this particular S protein has became the target of this our antibody generation. That's how we end up, and even using that cryo EM, the fine structure of this S protein into this actually tripart of different proteins, so that we can have a better antibodies targeting against this particular motif of the structure, has been the story of this antibody, the vaccine production. And this is some of the data of how this antibody interact with that particular the part of that spike protein of the coronavirus. But who cares? This must look too technical to most of you, so just forget about it. Yeah, so yep. So we can stop here for our first session. And then in the second session, we are going to go through the actual real the things about. But for today, we are going to go over only the prokaryote, the aspect, and then that will be the thing that is going to be covered for upcoming midterm exams. So beyond that, we are going to worry after the midterm exam.